यस्य रूपमधिकम् प्रच्छन्नगुप्तं धनम् विद्या भोगकरी यशः सुखकरी विद्या गुरुणा गुरुः विद्या बन्धुजनो विदेशगमने विद्या परं दैवतम् विद्या राजसु पूज्यते न तु धनम् विद्या विहीनः पशुः विद्या प्रशस्यते लोकैः विद्या सर्वत्र गौरवा विद्यया लभते सर्वम् विद्वान् सर्वत्र पूज्यते ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते।
Dr. Shilpa Jadhav is an eminent microbiologist with an extreme passion for teaching with firm belief in shaping society through education. She completed her master's degree in microbiology in 2012 from the University of Mumbai. Her doctoral research, which focused on microbial siderophores, earned her a Ph.D. in 2024, also from the University of Mumbai. With 12 years of dedicated experience teaching microbial biochemistry at the undergraduate level, Dr. Shilpa Jadhav has made a lasting impact on her students. She is a recognized teacher for M.Sc. Microbiology under the University of Mumbai and also serves as Ph.D. research guide in the same subject. Dr. Shilpa Jadhav has been an active contributor to academic development, serving on the syllabus-framing committee for M.Sc. Part 1 Microbiology at the University of Mumbai. Additionally, she plays a vital role as a theory and practical examiner for T.Y.B.Sc and M.Sc. Part 2 Microbiology examinations. Her research interests lie in applied microbiology with a specific focus on plant growth promoting molecules bioremediation. Dr. Shilpa Jadhav's scholarly contribution includes 10 research publications and a book chapter showcasing her communication to advancing her knowledge in the field of her choice. Through her expertise and dedication, Dr. Shilpa Jadhav continues to inspire students and contribute to growth of microbiology as a discipline. So welcome Vidyavachspati, Dr. Shilpa Jadhav.
Hello. The topic for my PhD is Microbial Siderophores, Production and Application. I have done PhD in the subject of microbiology from University of Mumbai, registered in 2008 in Bhavans College, Andheri, under the guidance of Dr. P.D. Anthapan. I completed my PhD work in the year 2011, post which I had my Viva in 2012 and I was conferred a degree of PhD in 2012. The topic for selection of PhD was Microbial Siderophores, a particular area which was actually not trodden in the Department of Microbiology, Bhavans College, and neither in any other colleges at that time in 2008, in Department of Microbiologies or Biotechnologies from University of Mumbai. So I decided to work on a particular area which was ideally a little new, a niche which was unexplored. And the most important part was I was trying to look for microorganisms and their applications in plant growth promoting factors, because essentially India is an agricultural land. So whenever we are trying to do a particular kind of work in PhD, my thought behind the work was that whatever I do as a part of my research should have application from lab to land. And that's why I thought that if I work in this particular area of research, it can have direct application into the lab and thereby can be brought into the form of a commercial product, as well as help the farmers to replace this particular kind of an organic product which was natural from microorganisms and can be used to replace the chemical pesticides as well as the chemical fertilizers, and thereby improve the fertility of the farms as well. So let us begin the journey of PhD. Microbial siderophores are ideally chemical ligands that are produced by microorganisms, and then they are excreted out into the external environment so as to chelate the ferric ions which are ideally present into the Fe+3 state and which are not in the assimilable form. What I mean here by non-assimilable form is that Fe+3 is abundant in nature, but it cannot be utilized by any kind of a living cell as a form of nutrient. What it requires to be done is that it has to be converted into Fe+2 state, which becomes assimilable. And for this very reason, microorganisms make use of siderophores, which will they will quench the Fe+3 molecules because they have a very, very high affinity towards the Fe+3 molecule. Then they will form a complex of siderophore as well as Fe+3, and this particular complex will have a receptor present onto the surface of the microbial cell. On the receptor, the complex goes and binds, and as the next step of the reception or chemical signaling, the Fe+3 molecule will be sent across the cell wall and the cell membrane. During this transfer or solute transport system, the Fe+3 molecule then gets converted into Fe+2 and then becomes available for microorganisms to be assimilated now that it is present inside the cytoplasm of the cell. So this is ideally the function of siderophores. They are the chemical ligands, they are organic in nature, very small in size, almost 600 Daltons, but have been also observed in a range of around 200 Daltons to as large as 20,000 Daltons, and they aid in the ferric transport and thereby making it available to the microbes. Now, why ferric transport is important? Because iron is one of the most important requirement for microorganisms to function into various varieties of cell function, whether it is physiological, whether it is cellular, or it is even metabolic. Most of the microbial pathogens will secrete siderophores because they require iron as an utmost requirement to survive and create pathogenesis. So if iron is not available, these pathogens will not be able to express the virulent factors, and as a result of which they lose their characteristics of pathogenesis. Besides that, iron is also important as a cofactor into the electron transport system as well as production of many enzymes in forms of the cofactors and coenzymes, and as well as certain types of proteins which we call as the iron sulfur proteins. The types of siderophores that are produced by microorganisms are essentially belonging to 4 particular types. The first is hydroxamate group of siderophore, the next is catecholate group of siderophore, next is carboxylate group of siderophore, and last is phenolate. So this is ideally the broad classification of the siderophores that are produced by microorganisms. Besides the kind of ion transport that the siderophores are involved into, we have also been seeing as a part of research that not only ferrous but certain other metals like copper, magnesium, manganese, which are also equally required as a part of growth factors, micronutrients by the microorganisms, are also made available by siderophores, because some of the siderophores that are secreted by microorganisms, they are not having only limited affinity to ferrous, they can also bind to some of these particular types of metals and thereby quench them, increase their concentration and make it available for the microorganisms as a part of their growth factor. I started my research work with literature survey, and over a period of this particular time when I was doing my literature survey, I came to know that microorganisms from terrestrial backgrounds as well as from aquatic systems are both known to secrete siderophores and excrete them into the environments. The very first thing that I had to start as a part of my PhD was isolating microorganisms from the different sources that I had decided on. So I had thought of terrestrial sources. I utilized 4 different soil samples, as well as I utilized 6 different aquatic samples which were collected from regions in and around Mumbai, and they were basically utilized as a source of microorganisms which can be utilized to produce siderophores. Objectives that were set for my PhD work was screening and procurement of siderophore producers from soil as well as aquatic ecosystems by primary and secondary screening methods. By primary screening methods, I meant to say I was doing selective enrichment, and post selective enrichment I was trying to quantify what are the types of quantity of siderophore that a particular microorganism were able to produce. The next objective was optimization of the growth factors using conventional method, where I basically selected carbon, nitrogen source, pH, temperature, presence of ferric at a specific concentration as the parameters under study, and post which I have also done comparative study on optimization using the statistical parameters like Plackett-Bourbon method and response surface methodology. Once the yield of the siderophore was established via the optimization studies, I isolated these particular quantity of siderophores and started utilizing it, or started doing application studies. So the very first application that I was trying to do with siderophores was to check for its biocontrol potential, wherein these particular siderophores which were made or secreted by Pseudomonas aeruginosa species, which I isolated from the soil sample, was found to be inhibitory against the fungal pathogens like Aspergillus niger and Fusarium oxysporum. The second objective that had been set for the PhD study was assessing the plant growth promoting ability of Achromobacter xylosooxidans. So this was also a microorganism which was isolated from a variety of soil, and the siderophore that was secreted by this particular culture was produced on a large quantity, and we checked its ability to increase the plant growth in comparison with the control systems. So coming to the primary screening, the samples that I had used was 18 different samples collected of soil as well as aquatic and rhizospheric locales. Soil samples from garden, compost, mangrove, Vinogradski column as well as vermicompost was utilized. Aquatic samples that were collected were swimming pool water, creek water, pond water, lake water, sea water and borewell water. In case of rhizospheric soil, the wheat field, rice field, spinach field, methi field, corn field and onion field soil was collected from the rhizospheric area. Rhizospheric area is ideally a particular zone where the roots of the plants are spread, and besides which there is a particular kind of a niche that is formed due to the secretion or the extracts of the plant which is exuded into the soil, and that secretion and exudates becomes a part of the nutrition for the microorganisms, and those particular zones of soils will be seen to be having a very high number of microbial activity, and thereby the proximity of getting microorganisms which has the ability of producing siderophores was very high, because whatever kind of siderophores were extracted and the iron was quenched, that would have been also made available for the plant as a part of its nutrition. The media that I utilized for doing the primary screening was selective enrichment using chrome azurol sulfonate broth. Chrome azurol sulfonate is a dye by itself which is ideally orange in color, which when added to the media with a pH of almost 7.2 has the ability of reacting with the ferric ions that are present in the form of FeCl3 in the media, and once there is a complex of the chrome azurol sulfonate as well as FeCl3, you will see the dye which was originally orange in color changes its colors to blue. So when we make the media, the color of the media is blue. Once the microorganism starts producing siderophores, these siderophores are secreted outside the microbial cell, so they become a part of the media. Once these siderophores reach the media, they will now start competing with the chrome azurol sulfonate dye because they have higher affinity to Fe+3, as a result of which the complex of chrome azurol sulfonate dye along with FeCl3 will break, and the siderophore will form a complex with FeCl3, as a result of which the chrome azurol sulfonate will take back its own color, which was orange, and as a result of which when we start the enrichment, the color of the media was blue. When we see the sample is getting enriched and has microorganisms which is producing siderophores, you will see the color of the media will start changing its color from green to yellow to orange, and thereby indicating that yes, this particular sample has microorganisms which has the capacity of producing siderophores. This particular enrichment was then isolated onto the assay media, as in the agar plates, and then the isolated colonies were picked up which were able to show us a zone or a halo around the colony which was orange in color. This way, after the primary screening, 108 isolates had been procured from 18 different samples. At the same time, for comparative study, we had also ordered a culture of Pseudomonas aeruginosa MTCC 2581, which is a marked siderophore producing organism. Further, for secondary screening, the organisms were grown in modified succinate medium for 24 hours at room temperature, and they were checked for the siderophore production capacity. The ones which gave us siderophore production in very quickly at a high concentration were then selected for further assay, and the detection and quantification of siderophore was done with the help of the Pines method as well as the FeCl3 assay for quantitative estimation. This helped us to screen out samples from 108 to further almost 10 good siderophore producers. Then these particular 10 siderophore producers were put for secondary screening, as I just mentioned, using the qualitative estimation by the FeCl3 assay, which is also known as the Atkins assay, as well as the Pines method, and thereby we selected out of this 10 cultures organisms. We scaled down to 2 particular organisms which were capable of giving us very high siderophore producing units, and thereby we started our further work with this particular kind of area. So generally from rhizospheric locales we had 34 cultures that we had isolated. From terrestrial samples we got 45, from water samples we got 11 isolates, and thereby once we had these particular isolates, from every particular type of sample we further did the secondary screening, and then we came down to 10 samples and further to 2 organisms which were known to be good siderophore producers. So with these 2 particular organisms which we had got from the cornfield, we started with the optimization studies, and the optimization of physicochemical parameters for enhanced siderophore yield was carried out using the conventional method. The very first parameter that we took under consideration or for optimization was influence of carbon source, where we made use of variable types of sugar sources like 1% glucose, xylose, maltose, sucrose, glycerol, mannitol, starch. So ranging from monosaccharides to disaccharides as well as polysaccharides like starch, and see its effect onto the yield of siderophore. The second parameter that we assessed were influence of organic nitrogen content and the percentage at which we use this organic nitrogen was 0.5% or 0.5 grams per 100 ml. The organic nitrogen source that we ideally tried were the peptone, tryptophan, malt extract, yeast extract, beef extract and soybean. Whereas the inorganic nitrogen source that we assessed was ammonium sulfate, sodium nitrate, ammonium chloride, urea, potassium nitrate and sodium nitrate. We also checked the influence of pH on the siderophore production, and the range of pH parameters that we assessed were from 2, 4, 6, 8 and 10. The influence of temperature was characterized using a range of temperature from 10 degrees Celsius to 55 degrees Celsius at a interval of around 15 degrees till we reach 55 degrees Celsius. Influence of aeration, where we maintained the siderophore production at static conditions as well as shaker conditions of 120 rpm per minute, and influence of ion concentration was assessed using a range of 10 microgram per ml to 40 microgram per ml at an interval of 10 microgram per ml respectively. We also checked the influence of amino acids like glycine, lysine, threonine, alanine and arginine at a concentration of 0.1%. This was the effect, this was the kind of conventional parameters that we checked, and the optimization studies were done. Further, we saw that both the cultures were put for these kind of conventional optimization studies. One was CW2, which was creek water, and the isolate number 2, which was further identified by 16S rRNA sequencing as Pseudomonas aeruginosa. And the other culture that we found to be good enough was CF1, which was cornfield 1 isolate, and we would see that it was capable of producing a good quality siderophore. Quantification was also very high, and this was further identified by 16S rRNA sequencing as the Achromobacter xylosooxidans. So after the conventional optimization studies, we could see that the carbon source which was helpful to increase the siderophore yield was glucose. There was effect of nitrogen, organic nitrogen source, where we could see where there was more of tryptophan that was added into the media. It could see influencing or enhancing the yield of siderophore production. There was no effect of amino acid concentration or any kind of inorganic nitrogen source on the siderophore production. Looking at the pH and the temperature, the optimum pH in case of both the organisms found was neutral, under which the enhanced siderophore production was seen, as well as the temperature was found to be 25 degrees Celsius, which was able to give us better siderophore production. Utilizing this particular data, further then we did optimization by Plackett-Burman method or the response surface methodology, which is a statistical tool that is available for optimization, online tool where you can make a particular kind of systems using many variables at a particular period of time. So here we could see we had utilized initially while doing Plackett-Burman, we had 12 different runs that were established utilizing pH, temperature, shaker conditions, incubation time, the presence of ammonium sulfate, the presence of peptone as well as glucose, as well as the concentration of FeCl3, whether it is having any effect onto the production of siderophores. And then we could see that yes, there were certain range in which the production was seen to be enhanced, especially having the pH to be neutral ranging from 6.5 to 7, the presence of glucose at 1% concentration, and at the same time the presence of ammonium sulfate was also enhancing the siderophore yield. So the kind of results that we got from the conventional method, we could see it was also seen by the statistical methods, and as a result of which we could very clearly propose this or mention this in our findings that whatever kind of increase in yield that we are getting was found to be statistically significant, and for this we also utilized the statistical test for finding the statistical significance limits under which this particular work was proposed. Once the yield of siderophore was enhanced, we could see that the initial yield in case of Pseudomonas aeruginosa by conventional method was 4.8 mg/%, which by statistical tools like the response surface methodology or the Plackett-Burman method became almost 12.8 mg/%. Whereas in case of Achromobacter xylosooxidans, the initial yield after conventional optimization was 7 mg/%, which was enhanced by the Plackett-Burman method to 15.9 mg/%. This way we could go ahead. We had already done the molecular identification of the cultures by sending these particular cultures to the 16S rRNA sequencing process. So now whatever kind of siderophore yield that we had to put it for application. So the siderophore that was extracted from Pseudomonas aeruginosa species identified by 16S rRNA method were used to study the antifungal activity by the plate method. So the antifungal activity was tested against the known fungal pathogens called as Aspergillus niger, NCIM 1025, and Fusarium oxysporum, which is NCIM 1008. The media that was used for the growth of fungal strains was Zepectox agar medium, and the pH was adjusted to 7, because if we would make the pH acidic, probably the siderophore function would have decreased. The antimicrobial activity was studied by the agar well diffusion method, and the siderophore samples that were put under study were undiluted, 1:2 diluted, 1:5 diluted, and 1:10 diluted into the system. The incubation conditions were RT or room temperature for around 48 hours, and the zone size of inhibition was checked as a parameter of assessment or results. So we could see that post the incubation conditions, we could see that yes, there was inhibition of both the cultures, Aspergillus niger as well as Fusarium oxysporum, by the undiluted siderophore sample as well as 1:2 diluted siderophore sample. But in case of 1:5 and 1:10, the activity decreased very rapidly, and this suggests that it's better to use the siderophores in a neat sample if at all we are trying to utilize it in the commercial application. So this kind of siderophore can very much be utilized to coat the seeds before germination, because if you are, for example, if we are planning to grow bajra or we are planning to grow jawar, these particular seeds, when are put for germination into the farm, they undergo infestation by the fungus which is present into the soil, as a result of which the overall yield of the plant basically suffers. So if we treat the seeds or coat the seeds with a particular kind of xanthan gum plus siderophore mixture, and we utilize this particular seeds, we could see that the seeds were not getting infected by the fungus, as a result of which there was a very high percentage of growth from the seeds, as a result of which the entire yield of the particular plant was very high, and thereby enhanced plant yield could be obtained from the farm. So this particular study had a direct application into the field. Fusarium oxysporum is a kind of a culture or a known pathogen which is known to cause the wilting of leaves. So the siderophore that was produced by Pseudomonas aeruginosa can also be used directly for foliar application or spraying onto the leaves, as a result of which it will not get infected by Fusarium oxysporum, and thereby the growth of the plant will be enhanced, and subjectively the yield of the plant will also be enhanced. Coming to the last part of the objective, which was experimental studies of siderophores that was isolated from Achromobacter xylosooxidans, and we were trying to assess its plant growth promoting factor, and this was basically tried onto the Trichoxanthus anguina, which is a plant, and the yield that we get from this particular plant is padval. So if you see padval in its natural form, then it is very light green in color. So the skin of padval is very light green in color, and thereby it very clearly indicates that the chlorophyll content of the skin of padval is very less. But padval has been seen to have medicinal effect into decreasing the glycemic index, as well as also having certain types of applications into certain kadhas which are utilized for respiratory infections. So I chose this particular plant, and it is also commercially grown on a very large scale in the farms of Maharashtra as well as throughout India. So this particular plant was selected. When we have set up this particular experimental studies, we had thought about 4 to 5 different treatments, but we scaled down only to 3 treatments. The first was control, which was just plain soil in which the padwal seeds will be grown, and thereby we'll get a plant, and thereby we'll check the yield. The second was the soil, which was fortified with manure of vermicomposting, which was weekly added onto the soil surface. And the third or the last or the most important parameter for our studies was soil carrier based culture, which was siderophore culture broth was applied weekly to the soil at the rate of 50 ml to the pot, which was carrying around 5 kgs of soil. So this way we started with our plant growth promoting factor experiment and the assessment parameters, where we studied the effect of siderophore during the entire period of planting on Trichoxanthus anguina was plant growth rate in terms of shoot length, weight of the plant, stem thickness of the plant, leaflets, the number of fruits every treatment, microbial content of the soil, the length of the fruit, the weight of the fruit, chemical content of the fruit skin, and chemical composition of the soil. So altogether 10 variable parameters we checked with the kind of experimental setup that we had for this particular study. So looking at the overall results that we got, the siderophore application definitely had a higher hand. The kind of results that we obtained of plant growth only in soil were very shunted. Compared to this, soil plus manure had better growth of the plant, but the best growth of the plant was obtained in the system where we had applied the siderophore producing culture broth to this particular pots as a part of the study. It had a direct influence onto the rate at which the plant was growing, the quality of growth in terms of shoot length, as well as the number of leaves that were formed by the plants which were treated with siderophores. During the entire growth process, we also checked for whether it was getting infected by any kind of fungi, the shoots especially, which was not seen in case of the plants which were treated with siderophores. Post which we saw that the flowering was also induced faster in terms of plants which were treated with siderophores. And finally, when we checked the fruiting or the fruits that were seen in this particular plants were definitely higher. So the number of fruits that we got in the case of system which was supported or applied with siderophores were definitely higher. So number of fruits were also higher. The length of the fruit was also longer. The weight of the fruit was also very high, as well as the most important parameter, that the skin of the yield or the padwal was also showing a very high chlorophyll content. So it had a direct influence on to the quality of the yield that was produced by the plant, and thereby it directly increased the medicinal property of this particular plant that can be utilized for whatever kind of preparations, especially maintaining the glycemic index and the kadhas in which these particular plants are utilized. This way my objectives for the PhD work was completed, post which I compiled it into a thesis, submitted it to the university, underwent the viva, and cleared my viva and got my degree. So in the entire process of PhD, it was not only enhancement of my academic profile, but it also instilled in me characters of being a good researcher, as well as certain kind of qualities which I have to develop as a human in terms of making persistent efforts, perseverance towards any kind of work that is taken over, and ability to work hard under challenging situation that ultimately leads to a very satisfying and experiential learning process. Thank you.
ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते न हि ज्ञानेन सदृशं पवित्रमिह विद्यते